It is my pleasure to welcome all of you, Dr. Karthik Raman, and all my colleagues and friends and students, and those who are going to join also in the next few minutes. I welcome all of you for the continued webinar series of the Chemistry Department. And it's my pleasure to host Dr. Karthik Raman, whom I requested about 2 or 3 weeks ago, and he readily agreed. He said this may not be directly a chemistry topic, but I told him that he could find that out as much as he would like to, but that's not the main requirement. The requirement is that the students of chemistry and the students of biotechnology will also have a chance to come together if a lecture is given, and it's possible for them to also interact. So I'm pleased to introduce Dr. Karthik Raman to all of you, who is currently an associate professor in the Department of Biotechnology, our own institute. And Karthik's general research group works on the development of algorithms and computational tools to understand, manipulate complex biological networks. He also coordinates the Initiative for Biological Systems at IIT Madras and is a core member of the Robert Bosch Center for Data Science and Artificial Intelligence, which is there next to his building in the new biotechnology building. I think the whole 5th floor is all Robert Bosch Center, right? If I understand.
Yeah, that's right, yeah.
Yeah. So a little bit about him personally, which I have taken from the bio that he has published. He did his schooling in the DAV Boys Senior Secondary School in Chennai and did his graduation from the Department of Chemical Technology in the UDCT then at the University of Mumbai in 1999, and he had a bachelor's degree in chemical technology and joined IISc Bangalore for the Masters in Computational Science. Later, he converted that to a PhD program. This is the standard MS PhD program of IISc Bangalore in Systems Biology. And he had his advisors as Dr. Nagasuma Chandra and Professor Saraswati Vishweshwara. His PhD was granted in 2008, and he was later in the lab as a postdoctoral research fellow of the lab of Professor Andreas Wagner in the Department of Biochemistry in the University of Zurich. And after finishing his postdoctoral work, he joined the Department of Biotechnology here in IIT Madras in 2011, and now where he continues as an associate professor. So besides this, he has a brief intro about his family. You can all look at this in the IIT Madras website, K. Raman. And his major interests, other than the subject matter, his other interests are computational linguistics for Sanskrit. Sorry, just one more. Linguistics for Sanskrit, numerical software, and semi-numerical algorithms. Wow. So you are with Donald Knuth's Volume 2. Okay, good. 1970s, 77, 78 Stanford, isn't it? Very, very early on. Yeah. And the founder of Tech and the later Leslie Lampert's LaTeX and so on. So this is associated with computer science and computational tools. And his hobbies are that he is an avid listener of Carnatic music and also likes singing and playing on the keyboard. So with these few words about him, I'm very happy to invite on behalf of all of you, Dr. Karthik Raman, to deliver a webinar on the topic of learning and predicting novel metabolic pathways through subgraph mining. Dr. Karthik, it's over to you.
Thank you. Thank you very much for the invitation to a webinar. So this is actually one of my first webinars after all the dozens of classes at the end of the previous semester. So I'm still learning the ropes on webinars, but I look forward to the experience. And so today I'm going to talk to you about learning and predicting novel metabolic pathways through subgraph mining. But before that, I thought I would give a quick intro to our group. I think Professor Mangal Sundar has already introduced what we do. So we work computationally, so in that sense we are actually surviving very well during the lockdown because our work has really not been disrupted too much, unlike many of my colleagues who require a wet lab. And we work on building scalable algorithms for studying metabolic networks. I think you're all familiar with metabolism. Basically think of them as a long list of chemical reactions that happen within every living cell. And we also look at in silico strategies for metabolic engineering, and we also perform a lot of data analysis using multi-omic data, so genomic data, transcriptomic, so basically data that stems from DNA, RNA, and metabolism, and so on. And we also, very actively drive the IBSE and RBCD side. And these are the key methodologies we use in our lab. There's something known as constraint-based modeling, and of course, quite a bit of high-performance computing, and a bit of machine learning. But the crux of, in fact, what I'm going to talk to you today is somewhere in this intersection between network biology and machine learning, and I will tell you quickly what that is. So the key research themes in our lab are, we focus on these techniques feed into several applications. So stemming from, say, metabolic engineering and pathway prediction, or trying to identify synthetic lethals in metabolic networks, designing gene regulatory networks, or most importantly, trying to understand and engineer microbial interactions. So this has been like a big thrust over the last 3 or 4 years, and we've had some very interesting work coming out there. And we also look at integrating multi-omic data and a lot of biological data analysis. And some ongoing projects in our lab, so trying to understand metabolic interactions in microbiomes. So you may have all heard of the gut microbiome and so on. I'll come back to it at the very end of my talk. Understanding redundancy and rerouting in these metabolic networks within every cell. Trying to understand the infant gut microbiota through metabolic modeling. And then what are the design principles underlying metabolic networks? What makes these metabolic networks tick? What are the key, design organizational principles? And how do you design these kinds of networks if you want to do it for synthetic biology? As suppose you want to design a new network, or you want to manipulate an organism to produce a new chemical, or to produce more of an existing important nutraceutical or something like that. And also we have been applying systems theory approaches. We're working with chemical engineers, trying to understand the structure of these networks. So I'll now give you a nice introduction to graph theory. So if you haven't heard this, I can assure you this will be the most entertaining part of this talk. So the history of graph theory goes like this. Very interesting. So the problem set in the picturesque city of Königsberg in 1735. Today it's Kaliningrad in Russia, around the Bregel River. The residents of the city had a question: is it possible to set out from my house, cross every bridge in the city, and come back home? Right? This was a very pressing question for the citizens of Königsberg back in 1735. And I hope you can see, it's a little tricky, but I hope you can see about 7 different bridges here, and there are, if you will, 3 different islands in the city. So suppose I were to start at this blue dot, and I have to cross all these bridges and come back. So this was the question that the city's residents had, and just like we start every conference or something with an invocation song to Lord Ganesha or Saraswati. So any math talk has to pay respects to Euler, because Euler has basically influenced every single field of math, and I typically discuss him more than a few times in my course in different, very disjoint topics arising from differential equations to graph theory. So what did Euler do? He wrote a very nice letter, so you should watch the language, of 1700s. Thus you see most noble sir. This was in response to the mayor of the city who actually wrote to Euler. The mayor of the city wrote to Euler saying, my citizens have asked me this question, and we are trying to figure the answer to this. Really interesting days, if you will. And he says, there's very little relationship to mathematics, and I don't understand why you expect a mathematician to produce it. For the solution is based on reason alone and doesn't depend on any mathematical principle. But then he, tongue-in-cheek says that even questions which bear so little relationship to mathematics are solved more quickly by mathematicians than others. And he said it's quite banal, but seemed worthy of attention because he couldn't solve it using just any of the established fields at that point. For example, using geometry or algebra or the art of counting. So what did Euler do? He took this sort of caricature map of the city and he converted it to this. Really speaking, he didn't draw this graph at that point of time. This is today's modern representation of what Euler did that day. So given this graph, would any of you be able to identify the walk the citizens were looking for? So for example, you have to give something like A, C, D, B, F, something like that. But you basically start at a node B, this circle B, this vertex as we call it, and you have to return back to B. And if you really, even if you look hard, you find that this is not possible. And Euler, in fact, proved that there is no, we name it in his honor today as an Eulerian circuit. Right? So you cannot start from any particular vertex in this graph, traverse every single bridge, and get back to the starting node. But really speaking, why are we worrying about it? Because this laid the solution of this problem, laid the foundations to the currently popular field of graph theory. And I'm sure even if many of you are not familiar with graph theory, all of you know social networks. Right? I'm sure we are on several social networks. So what are graphs? They're a very important theme in computer science and formally defined as G, V, comma, E, where G is the graph with vertices V and edges E, which consists of pairs of ordered pairs, if you will, or even unordered pairs of vertices from V. And they are typically referred to as networks. So you can think of road networks, social networks, metabolic networks, scientific citation networks. There are lots of people who study such networks to understand authorships, impacts, collaborations, and things like that. And graphs are classified elaborately, which influences the choice of algorithms. So you could think of directed versus undirected graphs, weighted versus unweighted graphs. So this may seem, a new terminology, but in fact, all of you have certainly used weighted graphs. Every time you open your Google Maps and try to find a path to your destination, you use weighted graphs, and typically Google will give you two weighted graphs. One that takes the shortest time, and one that is the shortest in terms of distance. So that is basically what a weighted graph is, and there are other kinds of graphs. I'll not go into them today. But some examples of graphs. So you're all familiar with many of these graphs. So like Facebook, Twitter, or at least, and of course there are protein interaction networks and so on. And every graph has this notion of a node and an edge. So what are the entities and what are the relationships between those entities? And if you see Facebook, the nodes are people and the edges are basically friendships. And if you look at something like Twitter, the nodes could be people or businesses and the edges are follows. And essentially you can see the difference between Facebook and Twitter. So if A and D are friends on Facebook, it's symmetric. So it's what we call an undirected graph. Whereas on Twitter, you might follow a celebrity, but they may not necessarily follow you back. And there are other kinds of networks. So we will talk about metabolic networks, or more importantly, molecular graphs today, where you can think of nodes as atoms and the edges as bonds. And I'll spend quite a bit of my talk talking about it. So why graphs? Because there are many questions that graphs can answer, and you'll be surprised to know that these questions were originally asked a lot by social scientists. And for example, do I know someone who knows X, the existence of a path? So how long is the chain to X? That's the shortest path problem. Is everyone in the world connected to each other? You may have seen the 6 degrees of separation and some interesting plays of that sort. Who has the most number of friends? Who's the most influential? In fact, you'll be surprised to see the applications this has. So there are, companies that try to figure out what are the least number of people I need to advertise a product to so that my product becomes popular or viral and things like that. Although viral is no longer an interesting word to use today. And can you predict if X and Y are friends? This becomes a link prediction problem. Right? So can you predict if two people are bound to be friends by virtue of their interests and things like that? This is how Facebook keeps popping up asking you if do you know this person, or do you think this person your friend? But that's for social networks, and of course I'm more interested in biological networks, and it turns out that the exact questions translate there again. Right? If you say I just changed the headings, the algorithms remain the same. So is there a way to produce a given metabolite X from a metabolite A? How long is that pathway from X to A? Are all proteins connected to each other by a path? And what is the most influential protein in a network? Maybe I want to find that and knock it out in a pathogenic organism. So I want to kill it by trying to take down its most influential protein. And can you predict if proteins X and Y are expected to interact? Right? So these kinds of questions one can ask. And there are many biological problems that map back onto graph problems, like pathfinding in metabolic networks, identifying important proteins, clusters of proteins, even the famed gene sequencing problem cannot be solved without graphs. Right? If you want to assemble all the reads that come from a next generation sequencer, you still need graph theory to do that. And there are some chemoinformatics problems as well, and one of them is what I'll sort of broach upon today. So this is so here is my talk finally. I've just given you the background on graph theory. So please do let me know if you have any questions on graph theory at this point. So this would be a time to just take a breather, and we can then continue with the rest of my talk. If you have questions, so please share them on chat or just unmute yourself and ask me the question. So this must remind you of, this is a classic question in JEE or the high school exam, saying I have this product on the left-hand side and I need to convert it to the product on the right-hand side. So what are the series of reactions that I must be employing? And typically, you have a very nice memory of organic chemistry. So you say I should use a particular, a Diels-Alder reaction, all these name reactions that we used to learn about. And can we use a combination of those reactions to convert a given molecule A to a molecule B? That becomes the question, and that is the exact question that we are trying to solve. So how do we go about it? So today we have metabolic networks for 100 plus organisms. These are basically, I'm talking about bacterial or even human and other mammals and so on. So there are people who've done lots of difficult biochemistry studies over the last several decades, and we now have a big knowledge base of several thousand reactions. In fact, there are tens of thousands of reactions, chemical reactions, known to occur in different living cells. And this could be as simple as glucose becoming glucose 6-phosphate, the first step of glycolysis in every living cell, or even much more complex reactions and very exotic reactions catalyzed by very talented enzymes, if you will. And these represent the varied repertoire of the biochemical conversions that the known enzymes can catalyze. And a lot of metabolic engineering today involves copy pasting these reactions, right? You take them from, say, a very a particular plant that is endangered, and you stick it into a simpler organism and try to see if you can produce the same metabolite. Maybe the plant is producing an important phytochemical that's has got anti-cancer properties or antioxidant properties, and you want to try to produce that with say E. coli or yeast, so that you can really scale it up and increase the yields and so on. But how do we predict these pathways? So, many a time, companies come and ask, I want to produce vitamin K27, or I want to produce riboflavin. So, and how can I make E. coli produce riboflavin? E. coli doesn't make riboflavin. So, how can I make E. coli produce riboflavin? So, can you trace a path that will start at, say some known compound in E. coli and take it all the way up to a new compound? And for example, can you do this for novel compounds that are not present in reaction databases? So there are, some compounds which are not yet, they may be drugs. So they've not yet, obviously they don't find their way into reaction databases, right? So our body doesn't make, say, penicillin. So so can you figure out how you can make such compounds? Or any other, say, lycopene or some nutraceutical or any of those compounds? And can you fill gaps in known metabolic networks? You typically find a lot of knowledge gaps. As people put together, assemble these networks, there are still a lot of knowledge gaps left behind. And even if you were to predict, can you rank these predicted pathways based on how plausible they are biologically? It's not simply trying to find that, okay, A goes to B, B goes to C. This is going to happen in biology. Is it going to be energetically favorable? Is this, a path that the cell would take at all? Are there other challenges, for the cell and so on? So many such interesting questions remain. So there are many tools that already do this. We weren't the first to do this. There are tools called RetroPath, PathPred, and so on. Metabolic Route Explorer is particularly nice. It's MRE. But nearly all of them rely on the existence of query molecules in the reaction knowledge base. So they look at the known molecules and then identify alternate paths or different paths or probable paths and things like that. Or they also rely on hand-curated reactant mapping. Meaning, there is a chemist or a biochemist who's looked at the reaction mechanisms and has identified what atom in glucose is now retained when it becomes glucose 6-phosphate and what atom has gone out into ADP and things like that. So these kinds of, which involves a lot of hand curation and a lot of manual effort. which means that this cannot readily scale to the size of databases that we have today. So we started out with this question. So can we build a fully automated engine to predict pathways between two molecules, relying only on structural information? You give me the structure of molecule A and you give me the structure of molecule B. I need to now predict a pathway not based on my memory of organic reactions, but trying to draw on what you have in the databases. What was our approach? So this is a simple diagram trying to explain our approach. We represent every molecule as a graph. So every molecule, I did talk about metabolic networks and I will talk about something like that shortly. But now every molecule itself becomes a graph wherein, every node, I'll have an example in the next slide, wherein every node is an atom and every bond is an edge connecting, the bridge of your Königsberg. And then we start with something known as reactant product mapping to identify the key transformation that happens. This is important, particularly in biology. Of course, it's, in chemistry as well. But you have typically a reaction A plus B going to C plus D. Actually speaking, the scaffold of A now remains in the scaffold of C. So A has become C, whereas B is an assisting molecule, a cofactor as we normally call it, which aids in the conversion of A to C. But the real action is happening between A and C. And C is the next molecule of interest, which you want to convert it downstream into some other molecule. And we did this mapping in an automated fashion using something called subgraph edit distance. I'll talk about it shortly. And then we did some subgraph mining to identify the reaction center, which is the actual site of action, the site of change, and some sort of neighborhood around that site, which we said is the signature that is necessary for that reaction to happen. Okay, so think of these as graphs and subgraphs, so subsets of molecules, like basically some neighboring bonds, right? So there is a carbon attached to a nitrogen, so this nitrogen, if it is attached to an oxygen, then this reaction occurs. Some rules like that we synthesize. And we identify such rules by summarizing what kind of transformations are happening, create a network where this is another network where the nodes are reaction rules and the edges connect rules that can be successively applied. I'm going to explain all of this. I'm just giving you an overview here. But the interesting point is, once we do this whole thing, our pathway prediction and ranking reduces to mere pathfinding on the reaction rule network. And pathfinding is a well-solved problem, as Google Maps keeps showing you. Here's your example. So this is D-lactic acid. So every vertex is an atom and every bond is represented by an edge. And we also have special edge labels for single bonds, double bonds, wedges, and the dotted, whether it's into the plane or coming out of the plane and so on. So now we need to mine reaction patterns. So here you go, there is a reaction. So it's a particular metabolite, you have some asparagine, aspartate converting amongst themselves. And when you have this kind of a reaction, you may be able to immediately spot that this molecule is probably becoming this molecule, and this molecule is very similar to this molecule. And these are absolutely side molecules that are important, but not mainstream in terms of what is the reaction. So we compare this as the minimum number of edits to convert a particular graph to another graph. So if I were to start with this, suppose this is becoming this, and I need to chop and add a lot of bonds and edges. So in fact, all of you are again familiar with this notion of edit distance, though not by the same word. It is what autocorrect does every day on your phone. Right? When you type out a message, it tries to find the words that are closest in edit distance to your mistyped word and suggest them as changes, or many a time autocorrect it much to your chagrin. So here what you have is subgraph edit distance, which is the same concept applied to graphs. So I have a graph. So you say that, 152 compound looks very similar to this 2631 compound, and that's what subgraph edit distance brings out in the end. And we identify this pairing. We identify the C42 is very close to C26 and C2631 is very close to C152, and we map these up. So once we map these, we also quantify the structural change. We say this is the reaction center. So there is this carbon where an OH is now being replaced by an NH2 is being replaced by an OH. So from aspartate to asparagine. So we take a pair of, and we find that as the reaction center. And then we look at the signature, which is basically the immediate neighboring an O here, an O here, a double bond, immediate reaction signature. And we find that there is a particular subgraph, basically a sub-molecule, if you will. So we have a molecule on the left that's converting to a molecule on the right, and you can see that this is where the action is happening. This is the reaction center, and there is an NH2 being lost here and an OH being added here. And then we see that there is a reaction signature. There has to be an O that is attached on either side, and there is probably a C here. So this is what is is the signature or the neighborhood where this action is happening. And then we store the structural change. So this loss of an NH2, gain of an OH at a particular carbon which has a neighborhood that looks like this. This becomes our sort of rule. So given this rule, see, right, for different examples. So for a simple example, suppose you have an alcohol dehydrogenase that converts ethanol to ethanal. So a C double bond O becomes a COH becomes a C double bond O, right? Your alcohol becomes an aldehyde. Right? So I'll have an example of this sort coming shortly. So for every reaction pair, for every pair of reactants which we mapped using our algorithm of subgraph edit distance, we store a rule that comprises the reaction center, the signature, the structural change, and also the cofactors or the helper reactants. And now we need to predict pathways. So given a target product molecule B, a rule L is applicable on B if L has a signature and you can add a subgraph to it and you find that subgraph in B. So this will become clearer with an example, and you basically can do it backwards. So from B, you basically remove the subgraph added, and then you add the subgraph removed from the previous on the other hand side to basically invert the reaction and find out what is the reactant you needed on the left hand side. It will become quite clear. So for example, here we have hexanal. So you need to remove the aldehyde group now and add the group that was removed originally in the reaction, right? The alcohol was removed and the aldehyde was added, right? And when you do that, you basically get hexanol, which was the parent molecule in this reaction. So, and essentially, this is how you can trace every reaction backwards and figure out a pathway from a given molecule A to a given molecule B. So each node in the network is a reaction rule, and edges connect rules that can be successively applied. So for example, if you have an edge from rule L2 to L1, the product of the rule L1 can be a reactant, successively apply them. And finally, the pathway prediction reduces to path finding on this network. So another example will make it clearer. Let's just take a very simple example where I have two rules which says that there is a reaction that can convert propanal to propanoic acid. So there is a CHO that becomes a COOH. There is some subgraph addition, removal, reaction signature. Let's not worry about it. And then you have a rule which says that ethanol becomes acetaldehyde. Right? So you have a COH that becomes a C double bond O. So given these two rules, can you now predict a pathway? Can you predict if there is a way to produce hexanoic acid from hexanol? Do you think there are enzymes that could do that job? And you can quickly infer that these would be the enzymes. Hopefully the enzymes that do these two reactions in sequence can do this conversion as well. But if you see, neither of the query molecules are in our reaction database, neither hexanoic acid nor hexanol. So you first search for a rule that is applicable on hexanoic acid, your target. And in our two-node network, L2 is applicable because it has the acid signature. Once you apply L2 on hexanoic acid, you get the parent molecule as hexanal. Because hexanal is still not hexanol, the source, you continue searching, right? You continue to search for your source molecule from the destination to the source. We are doing it reverse. And since L1 is collected from L2, we are guaranteed that L1 is applicable on the reactant produced by L2. And you apply L1 on hexanal and you generate hexanol as the reactant pair, and it is indeed the source. And your pathway is finally hexanoic acid from via L2 hexanal, L1 hexanol. I mean, I read the pathway out backwards because that is how we searched. But in reality, the pathway goes as hexanol, rule L1, hexanal, rule L2, hexanoic acid. So how do you do this pathfinding, right? So Google Maps, for example, does some a very simple pathfinding. It uses something called Dijkstra's algorithm. If it were an unweighted graph, you can even use a basic breadth first strategy. So I'll not go into the details, but we found that doing a simple breadth first strategy gives you longish pathways, and many of them don't rank alongside the biologically observed pathways. So what we did was we came up with a very simple idea, a heuristic. A heuristic is basically a nice computer science word for guesswork. So we said, let's optimize the edit distance to the source. So let's take a step towards the source, but don't make a major change to my molecule. Make a small delta change to my molecule, but in the direction of the source molecule. So this could be formulated as a best first search problem. And so this is a heuristic, and the motivation behind this was enzymes are very specialized. Enzymes don't do wholesale structural changes. And enzymes will cleave or remove a single bond, add a single moiety, and things like that. So that was the motivation for this heuristic, and it turned out this heuristic worked really well. We found that this particular heuristic consistently ranks natural pathways quite highly. We find that, here's a table which takes, in fact, central carbon metabolism. These are all some molecules from central carbon metabolism, and central carbon metabolism is very confusing in some sense because there's so much of interchange, transformation, and so on. The same molecule occurs in several reactions and so on. And we found that in a majority, our heuristic performed very well compared to many other known existing approaches, some very popular approaches. And in some of those cases, they couldn't even identify the paths between these molecules. And we also looked at what are known as the retrosynthetic pathways. So retrosynthesis is basically trying to find pathways to novel molecules. Say, can you find a pathway to a commercially important molecule like resveratrol or the anti-malarial drug artemisinic acid, and or even simpler things like tryptophan and histidine. We find pathways that are very similar to what is already known in nature or predicted in by other tools and in literature and so on. But most importantly, that is not the strength of our method. The strength of our method is to predict such pathways to unseen molecules, to molecules that don't exist in the database. If you see many of these tools like Metabolic Route Explorer or FMM, they basically predict pathways to molecules when the paths are already existent. But what we found was we looked for pathways to some unknown, some new interesting molecules. In fact, there's a very interesting set of molecules. These are three molecules that are implicated in tuberculosis. A very famous paper called The Breath of Tuberculosis. These are volatile organics that are present in TB patients. It is understood that it is synthesized by the TB pathogen, and we could actually predict these pathways to how these molecules must be produced inside the TB pathogen. And these compounds are not present in any of our training databases. We don't know any reaction to methyl nicotinate in current metabolic network databases. And of course, our approach does have some limitations. We don't consider thermodynamics, which could be quite, it was obviously very, very important. But the point is, our heuristic is trying to compensate for this to some extent. So we are able to still perform very well despite not considering thermodynamics. We are limited by the accuracy of the KEGG, but it's easy to extend our approach to other databases. We've been trying to work with some better databases than the KEGG. KEGG is a very popular database, but it has known erroneous reactions and so on. So that could mislead our algorithm. It's also somewhat difficult to predict longer pathways, but typically we don't worry about very long pathways because you cannot set them up in a new cell. You can't copy paste them into a new cell, into a cell and keep it stable. So to quickly summarize, we built a completely automated pipeline for pathway prediction and ranking based on a very efficient reactant product mapping that is fully automated, not reliant on expert curation. We identified reaction signatures, which basically represent the subgraphs necessary for a reaction to occur. And we then embedded all this information onto a reaction rule network, and all we had to do was just pathfinding on this network, and we could actually identify paths between unseen metabolites because we are only looking for the chemical signatures. So we also defined a new heuristic to perform this search, which tries to recover known pathways across organisms and enables a fairly biologically realistic ranking of these pathways, especially when you have hundreds of pathways. There are like many ways to convert between these molecules, especially in subtle carbon metabolism. Our approach is fairly accurate and quite scalable. We showed that, I didn't go into those details for today's talk, but we showed that even if you have reaction databases with maybe 100, I think 150,000 reactions, we are able to perform the predictions quite rapidly. The most important unique aspect of our approach is that we use no more information than just the chemical structures. So just using chemical structures, no other expert information, no thermodynamic information, we're able to give very good guesses on what is the most plausible pathway. And you can read our paper here, and our codes are also available if you want to run them from GitHub. So I just wanted to quickly touch upon other interesting things that we are doing with graphs, because I think there may be many people in the audience who are hopefully got an interest in graph theory at the end of this talk. So we've studied with Adi, we had a very interesting study on trying to see how mutations propagate in proteins. And it turns out the graph theory can be used to study protein structures as well. And then we've done some disease identification and disease module identification in different kinds of networks. This was a particular international competition, and which resulted in all the competition participants contributing their methods towards this very nice paper in Nature Methods. And we also had a very our method described in detail in this particular paper. And then we built a tool for enumerating all metabolic pathways in metabolic networks. This is not novel pathways, but just existing pathways, trying to trace them in metabolic networks. And we have exploited that to understand interactions in communities. I'll show you in the next slide. We've also used a very different application where we identify essential proteins in organisms using machine learning just from network information. And these are a couple of very interesting papers from our lab in the last, these two in the last couple of months, and this is a couple of years older. So wherein we enumerated a lot of these pathways and then understood how these pathways can be used to predict what kind of interactions happen between microbes, what is the level to which two microbes depend on each other from a metabolic lens. And how they such information can be used to identify very interesting interactions, right? So we find that the guts of some people recover much faster post-antibiotic treatment than some others, right? And this is because there are some very interesting metabolic underpinnings to this. So these are all rooted in graph theory. So I just thought you would find it interesting. So I'll quickly talk to you about our initiative on biological systems engineering at IIT Madras. It's an interdisciplinary group, and where we are trying to interface with the existing strengths in quantitative science at IIT. And our key themes are biological network analysis, as you can already understand, machine learning and systems theory. We have quite a few faculty associated with the center. So we have core faculty from chemical engineering and computer science, and of course biotechnology, and associate faculty from, again, chemical engineering and biotech. And again, a very interesting set of projects we are working on, trying to predict essential proteins in networks. The dream challenge I talked to you about, there are folks trying to investigate the role of gut microbiome in autism, and trying to identify the context of mutations in cancer. So all using data science and multi-omic data and so on. And also studying protein folding pathways or predicting preterm births, a very interesting project with an important clinical outcome. Especially in the context of India. So Arvind was the student who did a lot of work on this project. He was a dual degree student from computer science. And Shayan was faculty in computer science. He's now unfortunately moved to IIT Delhi and was funded by IIT Madras. So this is our lab, and I'll be very happy to take questions.
Thank you.